Hello, everyone, and welcome to today's webinar, Inspiring Credibility, the Benefits of Automation in Experimental Research. I am Marie Stone of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Leica Biosystems. To learn more, visit leicabiosystems.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, April Schrank Hacker, Ed.D, MSOD, CG, ASCP CM, Life Sciences Marketing Leader at Leica Biosystems. April, you may now begin your presentation.
Thank you for that kind introduction. Hello, everyone, and thank you for attending this talk about inspiring credibility, the benefits of automation in experimental research. I am April Schrank Hacker, and I am the Life Science Marketing Leader at Leica Biosystems. As a certified cytogenetics research specialist, I have spent most of my career on the bench working in translational research, assay design, and development. In today's presentation, we will talk about the importance of building credibility into your research. We will discuss reproducibility in terms of the 3 R's: reproducibility, repeatability, and replicability. We will help define what they mean and how they influence research studies. Finally, we will share a few tips and tricks about how researchers can leverage automation and inspire confidence in results. As we talked about earlier, if you have any questions, please add them to the chat, and we will have time to dig into them at the end of the presentation. Great, let's jump into it. A recent Nature paper shared results from a survey of over 1,500 researchers in which they questioned the reproducibility in research. The results were startling. 70% of respondees reported being unable to reproduce another scientist's experiment, and more than 50% reported being unable to reproduce their own. I for sure have experienced both of these predicaments during my research life. Have you? I bet each of you has one particular experiment that if I handed you the microphone, you could still tell us in vivid detail about all the times you reran that assay. For some reason, you could just never get it to work quite the same way. And for me, it created an unsettling crisis of confidence in the reliability of research. But hold on to that thought. We will decode some ways to inspire credibility in research as we go forward in the presentation. As we know, there are many ways to think about reproducibility as there are branches of science. But be careful, not all terms about reproducibility are created equally. A few key publications were instrumental in helping researchers define and refine what reproducibility means and how we can apply it. Marina A. Barba at the George Washington University published Terminologies for Reproducible Research to help us grapple with the conflicting definitions of what researchers and publishers use to describe what reproducibility is and how it is interpreted within the different sciences. The key idea is that the definitions mean different things to different groups. Research typically classifies reproducible results using a decision tree. Either A, authors do not distinguish between the words reproduce and replicate, or B, the terms are used distinctly and are clarified with subcategorization. Group B1 uses reproducibility where the original researcher data regenerates the results, and Group B2 collects new data to arrive at the same result as the previous study and refers to this as replicability, or the ability to repeat a study. The Federation of American Societies for Experimental Biology published recommendations targeted at biomedical and biological research, providing definitions that fall squarely into B2. So let's talk about how we can use the 3 R's, reproducibility, repeatability, and replicability to inspire credibility in research. As we discussed, reproducibility has similar or nearly identical results using comparable materials and methodologies by independent researchers. For example, looking at the liver sections to the right, each was generated from the same exact block, but stained at 6 different labs. We can easily see significant variability in the staining intensity and differentiation in each image. Lack of optimization of process can lead to difficulties with reproducibility. Repeatability measures the variation in measurements taken by other researchers under the same conditions or settings. In this image to the left, we can see several different tissues stained with similar consistency in each row. Using automation can impact repeatability. Replicability is the ability to duplicate or repeat a prior result using the same source materials and methodologies. It is important to think about that when you're referring to repeating the results of a specific experiment rather than an entire study. With the increased focus on rigor and reproducibility, the National Institute of Health and publishing groups Nature and Science held a workshop with journal editors in basic and preclinical science journals to investigate how to ensure rigor and reproducibility in research publications. A few details included having a good research design and elements such as complete methods and material section, replicate studies in range of conditions, and data reporting were identified as the framework for what good looks like. Following these guidelines can help others to repeat the experiment and speed therapies. Now that we've talked about what reproducibility is, let's turn to how we can use it as a framework and what tools, such as automation, can impact inspiring credibility in research. I propose automation has 4 supporting pillars or ways for potentially impacting research. The first is automation helps to generate trustworthiness in research. It also offers reproducibility through workflow optimization and standardization, optimization of workflows through instrument processing protocols, and together, these significantly inspire confidence in results. Watch for the color-coded icons throughout the remainder of the presentation to highlight the potentials for automation's impact on research. Let us define a few keywords that we will be talking more about when we talk about reproducibility. Trustworthiness is the credible, transferable, confirmable, and dependable. Optimization is operational efficiency and standardization of procedure. And confidence is how sure you can be of research. Some current methods rely heavily on manual processes that carry an inherent risk for inter-operator process step variability. Handwriting identifiers on slides can be transposed or illegible. I might dip my slides for 32 seconds, while my coworker might dip faster or slower. My side might sit in hematoxylin for 3 minutes, while the next one might sit for 5 minutes while the phone rings. We may have all experienced some of these examples in our daily lab life and know it's these tiny, hard to control for nuances of manual process that can significantly impact results. Automation removes human variability, which optimizes your workflow and can lead to credibility in your research. Keeping the 4 pillars of using automation to inspire credibility in mind, I'm going to invite you to rethink some of the current methods perhaps and share a few tips and tricks about how automation can impact research. Remember how variable our examples of manual tasks in the lab could be? Automation lowers the intervariability of different runs over different days and intra-assay variability, providing consistency between slides within one run, for example. The key is, automation offers confidence and consistency over repeated ones, which can positively impact research's building trustworthiness, reproducibility, and confidence. Manual process has variations along the workflow. For instance, each scientist or technologist has individual processing ways that even the best SOP can't control for. Other factors, such as time and temperature, can flux in manual processes, and even reagent preparation can introduce variability. We all know that scales are not created equal either. The image to the left is the same section of tissue, stained on different days, showing variability between staining runs. Automation can offer building consistency by monitoring sample conditions and applications, impacting research in terms of reproducibility. Automation supports reproducibility and drives confidence by completely removing manual processing and providing results through consistent handling of materials time after time. Looking down the image columns at the right, you can see how automation has consistent results on different runs in different tissues. Slide 1 can be equally assessed to 800 and 800 to 1600. Providing and reviewing protocol metrics is a critical success factor identified as a part of good research design. Automation provides onboard metrics capture for producing or repeating reliable results. Examples include protocol specifics, run times, specimen identifiers, reagent monitoring, and problem logs. Providing accurate and complete documentation in publications and in your daily work help in supporting trustworthiness and build confidence in research. The use of automation can improve efficiencies, optimize labor-intensive and error-prone manual processes within the workflow. To help you reimagine this idea, observe your current workflow over a few days, benchmark the current standard processing times, and the number and complexity of manual steps. Note potentials for intra-operational and inter-operational variability. By minimizing the human variable and removing opportunities for human error, automation encourages reproducibility, repeatability, and replicability, and inspires credibility. I remember furiously scribbling notes into my lab notebook, trying to capture every critical detail of the experimental steps in my protocol. As you can see, Marie Curie had the same process back when she was discovering radium. Today, automated instrumentation can be your partner in producing and tracking critical data, such as recording process steps during a run, what reagents you used, how long, and time in terms of how long did things run for, and temperatures. Also documenting protocol parameter data. Optimization by automation, by automating your workflow, can add consistency and reproducibility to your research. Why optimize workflow through automation? Foundations first. Automation can be a powerful tool in the lab, but be careful. Think about reproducibility in terms of a copy machine. They don't understand what they're copying. They just copy. Optimized workflows through automation can replicate quality results, but non-optimized workflow using automation will replicate suboptimal results consistently. We've talked about a few frameworks for inspiring credibility in research and a few tips and tricks for how automation can impact research. As a wrap-up, here are 3 easy steps to inspire confidence and credibility. First, reproducibility of results over time. Second, optimization of automation for operational efficiency. And third, and one of the most important, is trustworthiness. And not just within the research you're doing, but also within the field as an author that you're providing to other researchers, your affiliation, the university or institution that you're publishing on behalf of, and the publisher themselves, the journals and that you're working and writing through. I wanted to share a last resource with you. This is called Knowledge Pathway. Knowledge Pathway is our educational portal at Leica Biosystems where you can go and read content and learn more about building confidence in your research. Thank you for the gift of your time and attention, and I will be happy to answer any questions.
Thank you, April, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, what things should I look to include when thinking about publishing in terms of reproducibility?
Thank you. Well, that's a great question. So some of the things noted from the NIH workshop were a focus on rigor and reproducibility, such as developing a good research design and including complete materials and methods sections. Also, a descriptive statement of the replicate studies and accuracy of reporting data would be really great in the design of that.
Thank you. Next question. How does automation support ways to potentially impact research?
Yeah, so I really like to use the 4 pillars that we talked about. By building trustworthiness and generating credible and transferable methods, optimizing the workflow by having standardized instrument process protocols that make the data really easy and reproducible. And by doing all those things, it really inspires confidence in the results.
Great. Next question. How can automation help with reducing variability in my experiments?
I would say that automation really removes that human variable that comes with a manual process when you're doing your samples. It offers consistency run after run, and it really lowers the intervariability between runs over different days and even the intra-assay variability within one run.
Great. It looks like we have time for one more question. How can open source hardware help?
That's a really good question. I think there's lots of options that are now available to us the more automation becomes prevalent. I think one of the great things that you can do in terms of open source hardware is to really go back to your application specialists and ask them how that might apply to your application.
Great. Well, thank you again, April, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Leica Biosystems, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.